Hello everyone. I'm very glad to present my work AlignGraph. It is an algorithm to improve DNA-seq assemblies guided by closely related references. My name is Bao. I'm from University of California, Riverside. At first, I will talk about the background a little bit so that everyone will be in the, then I will introduce terminologies so that everyone will be in the same page. And then I will talk about different assembly approaches, de novo approach and the reference-assisted approach. Then I will show you the novelty of AlignGraph. And in the next part, I will talk about the AlignGraph algorithm in more details and show you evaluation results and finally make conclusions. So let's see the introduction part first. Next generation sequencing machines can generate short trays from a target genome. So here, the target genome means the genome from closely related, the genome to be sequenced. And assembly algorithms can assemble short trays into contigs or scaffolds. There are basically two assembly approaches. One approach is the de novo approach, and the other approach is a reference-assisted approach with the reference genome. So here, the reference genome means a genome from a closely related species. So it is different from the target genome. And currently, the length and the completeness of the contigs or scaffolds are still not so satisfiable. And there is a large space to make improvement. And we do have some opportunities. One opportunity is we have more and more newly sequenced genomes available, and we can make use of the new genomes and improve the genome sequencing. And AlignGraph is an algorithm using the reference genomes, and it is essentially a new method in the reference-assisted category. So let's first see the de novo assembly approach a little bit. The de novo assembly approach uses Bruijn graph as the underlying data structure. So what a de Bruijn graph is, it is a directed graph with one vertex per k basis. So in this example, we have two rays R1 and R2. We can construct a vertex TTTA from the first four bases of R1. And we can construct another vertex TTAG from the second four bases of R1, and so on. And we connect two vertices if they are continuous k-mers from the same read. So we connect these two because they are the first four bases and the second four bases of R1. And we also connect these two, and these two. We join two vertices if they are the same k-mers. We join these two because they are both TTTA. We join these two because they are both TTAG. And for the join, there are two cases. In case one, if R1 and R2 are in the same genome region, then the join would result in such a linear substructure. This is good because this makes the de Bruijn graph continuous. However, in case two, if R1 and R2 are in different genome regions, this join would collapse different genome regions into one single path, and this makes the de Bruijn graph hard to be traversed and may result in short contigs. So the second case should be largely avoided. And there has been actually several publications to avoid the false positive joins of the vertices. One publication is the paired end de Bruijn graph. The idea is to incorporate paired k-bases into each k-mer so that the additional information from the paired k-bases can help to avoid false positive joins of the vertices and can avoid branches. And this work is basically in theory. And another work is the positional de Bruijn graph. The idea is to incorporate alignment position into each k-mer. And this work is basically for correcting contig errors instead of directly for assemblies. And let's see the second approach, reference-assisted assembly approach. The idea is to use a reference genome to generate assemblies. And there are basically two methods. One method is to assemble rays from scratch into contigs or scaffolds. And the other method is to order and join pre-assembled de novo contigs or scaffolds with the undecided basis. So this work is basically a scaffolding, this method is basically a scaffolding algorithm. So actually, in the previous presentation, regopt is also a method, algorithm in this method. And essentially, AlignGraph is a relatively new method to extend and join pre-assembled contigs or scaffolds with decided basis. So it is different from the first method because it uses the pre-assembled de novo contigs or scaffolds, and it is also different from the second method because it makes extensions with the decided basis. And the only similar work we can find is from Ganier in 2009, and that work is for Sangaris. So AlignGraph is a relatively new method. And for the algorithm itself, there are 3 innovations. One innovation is it is a secondary assembly method, and another innovation is it is based on paired and multi-positional deep brain graph. And the last innovation is it aligns reads to the reference genome with the guidance from pre-assembled contigs, actually also scaffolds with limited gaps. So this is the assembly scenario. We have a set of reads. We assemble the reads into contigs, and then we use AlignGraph as a post-processor to join and extend the pre-assembled contigs. So here are 2 questions. One question is, why do we use the deep brain graph-based method? Why don't we just make heuristic extensions? And the other question is, why do we use paired and multi-positional deep brain graph? Why don't we just use the conventional deep brain graph? So here are the answers. Let's see, for the first question, let's see figure A. In figure A, we have a pre-assembled contig C0. We want to make extensions, and C0 corresponds to 2 repetitive regions in the genome, and we can extend it with either C or A. So there is ambiguity, and we cannot make extension because we don't have enough information to resolve the ambiguity. And this ambiguity is actually frequent because the reads are short and genomes are usually repeat-rich. And let's see the second case. If we construct a deep brain graph with the reads and the pre-assembled contigs, then such ambiguity is corresponding to such a branched substructure. And we have all the genome information incorporated in the deep brain graph, and it might be possible for us to resolve the branches and make extensions. So the deep brain graph-based method can help us to make more extensions, can help us to resolve more ambiguities. And the second question is, why do we use paired and multi-positional deep brain graph? The answer is very clear if we compare figure C with figure B. Because in figure C, the corresponding substructure with this branched substructure is actually linear. So we can easily make extension without having to worry about the branches. So this is why we use paired and multi-positional deep brain graph. And here comes the question, how the paired and multi-positional deep brain graph can help us to remove branches. I will talk about this later in more details. So let's see the AlignGraph algorithm. The first step is to align the reads and the contigs to the reference genome. So we align the reads directly to the reference genome with bowtie 2. Bowtie 2 is a highly tunable aligner. So we can use relaxed parameter settings for good sensitivity, and we can use paired and read alignment for good precision. And here the problem is we cannot handle large difference between target genome and the reference genome because the reads are short and the difference might be very large. So our idea is to align the reads to the pre-assembled contigs and then align the contigs to the reference genome. The contigs are long, so we can align the contigs with respect to the difference between the target genome and the reference genome, and the reads can be aligned accordingly. So this is an innovation, as I discussed before. So after the read alignment, here it comes the paired and multi-positional deep brain graph. So essentially, for the deep brain graph, for this deep brain graph, each k-mer is a 5 tuple containing the left k basis of paired and read S and the alignment position of S to the pre-assembled contig, PC, and the alignment position of S to the genome, PG, and also the paired K-basis alignment position to the pre-assembled contig, PC prime, and the paired K-basis alignment position to the reference genome, PG prime. So we have a lot of information incorporated in the k-mer, and we have more stringent requirement to join to k-mers. So the requirement to join to k-mers are the k-basis should be similar enough, and the alignment positions should be close enough. So we have more stringent requirement, and we expect to avoid a lot of false positive joins and avoid branches. So essentially, paired and multi-positional deep brain graph is a combination of paired and deep brain graph and positional deep brain graph. Here is an example to show the advantage of paired and multi-positional deep brain graph over the positional deep brain graph. Positional deep brain graph was introduced in my introduction part. So we have a reference genome in hand, and we want to assemble the target genome. So the target genome is unknown. And we have a pre-assembled contig. The contig is not complete. It misses target genome regions A and B. And it has regions C and C prime both aligned to C. And compared with the reference genome, the target genome has 2 copies of A and 2 copies of C. And the reference genome has only 1 copy of A and 1 copy of C. So if we let the k-mer, k-basis plus the alignment position to the reference genome, this deep brain graph would be a positional deep brain graph. And we align the reads directly to the reference genome. We don't use the pre-assembled contigs to guide the read alignment. Then many reads cannot be aligned, and the deep brain graph would not be so complete. So this is the resulting deep brain graph. It is a positional deep brain graph, and it is not so complete because a lot of reads cannot be aligned. And we have to use the pre-assembled contig to guide the read alignment. So more reads can be aligned, the deep brain graph can be more complete. However, here the problem is k-mers from target genome region A and A prime might have the same k-basis and alignment position to the reference genome region A. So they may not be distinguished and forced positively joined and result in branches. And similarly, k-mers from C and C prime might also be forced positively joined and result in branches. So this is the deep brain graph. The blue path corresponds to newly added path to make the deep brain graph complete, and the red path corresponds to forced positively joined vertices, and they result in branches. So there are 2 branches, 1 branch on the left and 1 branch on the right. So we want to incorporate more information to remove the branches. So we incorporate the alignment position of the k-basis to the contig. Therefore, the k-mers from target genome region C will have alignment position to contig region C. And k-mers from C prime will have alignment position to contig region C prime. So the k-mers from C and C prime can be distinguished and the branch can be resolved. And this is the resulting deep brain graph. The branch on the left in the previous deep brain graph is resolved. So there is only 1 branch left, and we further incorporate paired k-basis alignment position to the reference genome. So k-mers from target genome region A will have a paired k-basis alignment position to reference genome region E, and the k-mers from A prime will have a paired k-basis alignment position to reference genome E prime. So they can be distinguished also, and this is the resulting deep brain graph. It has 1 single path, and we can generate a long contig as is the extension of the initial contig. From our experience, we can achieve up to 5% better performance with this novel structure compared with the positional deep brain graph. So in the last step, we remove errors from the deep brain graph and traverse it and generate a contig as the extension of the initial contigs. So actually for the error removal, it is pretty easy because paired and multi-positional deep brain graph can not only avoid branches, but can also avoid false positive path and incomplete path. For false positive path, correct reads are aligned together with the reads with errors. So the errors can be corrected. And false positive path can be largely avoided. And for incomplete path, because we have the support from the reference genome, we can complete some of the incomplete path compared with the conventional deep brain graph. So we can get long contigs, so we can make extensions. So let's see some evaluation results. Actually, there is an evaluation software distributed with the AlignGraph software called EVO AlignGraph. The idea is to align the contigs to the target genome with blood. So the target genome is the ground truth, as I talked about in the introduction part. If a contig can be aligned with a global alignment, then it is a true contig. And if a contig can be only aligned with several local alignments, then each local alignment corresponds to a true contig. And our sensitivity and precision measures are both for true contigs. So we basically have three major sensitivity measures: the number of extendable contigs, N50 value, and the number of covered bases. And we have two precision measures: misassemblies per million base pairs, and average identity between contigs and the target genome. So we assembled reads from three species: human chromosome 14, Aetheriana, and L. erecta, into contigs or scaffolds with different assemblers, and we applied AlignGraph to the pre-assembled contigs or scaffolds to make improvements. So here are the results. These are the target genomes, these are the reference genomes, and we assembled with different assemblers into different data sets. So let's see the results. We can extend 11.6 to 62.3% of the contigs, and compared with the extendable contigs, we can improve the N50 value by 9.4 to 165.8%. We can also improve the percentage of covered bases by 3.4 to 29.0%. This means the alignment algorithm can effectively improve the sensitivity of the upstream assemblies. And let's see precision. Actually, we expect an increment of misassemblies and a decrement of average identity. But from our results, we found that the change of precision is in a reasonable range. So the increment of misassemblies and decrement of average identity should be reasonable. And this figure shows the human chromosomes 14 assemblies with the different reference genomes, from chimpanzee to macaque, from the highest similarity to the lowest. So along with the drop of the similarity, the percentage of aligned reads drops, and accordingly, the percentage of extendable contigs and the percentage of improved N50 value both drop. From our experience, as long as 50% of the reads can be aligned to the reference genome, we can achieve solid improvements. And actually, the performance of AlignGraph is highly dependent on the upstream aligners. So if we have a better alignment algorithm, we can align more reads to the reference genome, and we can achieve better performance. And we are working on this to design a better alignment algorithm with respect to the difference between target genome and the reference genome. So finally, conclusions. For the performance, AlignGraph can make solid improvements to the upstream assemblies, and it can work in combination with different assemblers, and we can achieve better performance with a better alignment algorithm in upstream. And the method is actually novel. It extends and joins pre-assembled contigs or scaffolds with decided bases. And for the algorithm itself, there are also three novelties. It is a secondary assembly method, and it uses paired and multi-positional deep brain graph. And the alignment is context or scaffolds guided alignment. So if you are interested in it, you can download it from this website for free. Okay, thanks.
So, hi, thanks for a nice talk. I have two questions. First question is, how hard is to make your tool work with another read technologies, like, for example, PacBio?
Means some other reset?
Yes, like,
We haven't tested with some other reset. We just use Illumina reads.
okay. And other question is, there is usually one more useful measure for assembly qualities, N50 corrected, like you just break the contigs at each error and you just take the N50 of that. How does your assembly fare in that measure? You don't have...
You mean the change of precision?
Yeah, like you do introduce much more errors, or? Okay, so you introduce fewer errors.
So sometimes we have decrement of the ISMB value, but actually sometimes we may have a little bit better ISMB value.
Okay, thanks.
Yeah.
Hi. What's your experience on using this tool with on a gene family? So have you assembled gene families by using a reference such as a human reference and then trying to assemble like a monkey or something?
You mean use different reference genomes?
Yeah, different reference genomes. So example for gene families, it's often that the regions are homologous and the genes are sometimes close to each other. And so it's often difficult to distinguish, when you're doing a de novo assembly, it's often difficult to distinguish those genes. And I mean, have you had any experience in assembling those regions with this tool?
So we don't actually have the quantification, but the experience is the closer the two species are, the better assemblies we can achieve. So from the chimpanzee genome to macaque, the similarity to human drops. So we got smaller improvements.
Okay, thanks.
Mm-hmm.